Hi, my name is Sophia McNamara. I'm a senior at the Telluride High School. And this summer, I studied microbial ecology at the Schmidt Lab at UC Boulder. So my mentor was Dr. Steve Schmidt, but I also worked pretty closely with postgraduate students and undergraduate students. So our experiment was basically studying the effects of nutrient addition to find the limiting nutrients for cryoconite holes, which are basically sediments from glacier in Antarctica. And here's some pictures from the lab. Here's like that chemical section, and then the experiment actually already began when I got there. So here's some pictures of the samples inside their incubator. So a huge part of my internship was experimental maintenance. So I spent a lot of time making sure that the temperature and light conditions were consistent to those in the natural Antarctic habitat within the incubator. And we also made sure that the water content of each sample was adequate. So we weighed it to be sure. And then another huge part of it was counts, which I'll get into on a slide a little bit later. So here's some pictures just of the data. There's some light and temperature data. We recorded everything in a notebook and then put it into something like Google Sheets or Kaleidograph to keep better track of it. And okay, so counts are what we did to quantify the microbial data. So if you look at that graph over there and look at the shaded area, we would look at that graph underneath a microscope, and when a certain microbial growth, like NOSDOC, would intersect with that shaded area, we would count it as a hit, and in that way we could approximate the percent cover of a certain plate or a sample group. So throughout my experience at the lab, I became pretty familiar with lots of equipment. I got to play around with different microscopes of varying powers, and I became familiar with a qubit, which is basically what we used to quantify the density of RNA extractions. I became familiar with lots of different types of sterilization, UV, chemical, and autoclave sterilization, and I became familiar with lots of computer software to organize and quantify the data. So a big part of my internship was looking at data from a previous Svalbard experiment. It was very similar to the one we were working on, but it just used sediments from Svalbard. And my mentor, Steve, thought it would be a good idea for me to look at this and run some statistics tests on the data. So, next slide. There we go. I ran a multiple variable statistics test on the data and wrote up a little report comparing the different nutrients and how they interacted with the microbes in the sediments. And then next slide, sorry. And then I graphed the growth over time using the percent cover I talked about before. And I tried to find a mathematical expression that would approximately fit the data, and I ended up finding that a logistic equation would fit the data the best, and I was able to compare the intrinsic growth rate, or R, in the logistic equation between the different sample groups, and I found that my conclusions were pretty consistent with the statistics test that I had run previously, just looking at the data straight on. So a huge highlight for me was microscope photography. Over here, that orange growth is NOSDOC, and these are about 40x power, just looking at the samples like in a more everyday microscope, what we would use for counts. And then I got to look through a compound microscope at 400 to 1000x power, and this was probably my favorite part of my whole internship. These are the same grows that we were looking at on the lower power, but just super magnified. You can see the chloroplast within the cells, which is pretty amazing. And then this horseshoe shape is a diatom, which is a single-celled algae that has really striking geometric patterns, and that was just awesome to find. So outside the lab, I spent a lot of time hiking around Boulder, running, exploring downtown, and spending a lot of time with my wonderful host family. I think I had the same host family as TJ, so Eben, Jean, Shay, and Quinn. They were amazing. And my advice for future Pinterns is to don't be afraid to make work for yourself. There's going to be days where maybe you don't feel like you have anything to do, and those are the days where you go up to your mentor and ask, oh, hey, what can I do to help out? That's how I got involved with the previous Svalbard data, and I would have never done that if I hadn't gone up to my mentor and asked for something to do. And overall, a very helpful and fun group of people in the lab, and I'm so grateful. Next slide. I'm so grateful for everybody that made this possible.